these tutorial quizzes. And Dr. Pemberton is available for office hours for tutorial material. So Dr. Pemberton will be in his office, which is BGS 3030. So he's available every Friday from 2 to 3. If you can't make it at that time, email him and see him another time. Okay, but the main thing is that you guys have a test this upcoming Friday online from 8 a.m. to 6 p.m. covering those 4 lectures. All right, so the exciting world of the cytoskeleton. So we were talking about tubulin, and you have this tubulin dimer. If you go above a critical concentration, you polymerize, and if you have a nucleus, you polymerize faster. Hopefully that all makes sense. Now, putting in the context of a cell, the cell wants to regulate things. The cell has evolved to regulate things. So yes, if you're above critical concentration, you will polymerize. Yes, if you have a nucleus, you'll polymerize faster. You'll get to elongation faster. So the cell just regulates how this is happening, right? So where are microtubule dimers going to be above a critical concentration? Wherever the cell translates them. So the cell has ribosomes, it does translations in different locations. So it will translate things where it wants things to occur. Wanting being human, but it has evolved that way. There's an advantage to the cell doing certain things. So when you look at the centrosome, which is the microtubule organizing center, the cell has put things in that location. So in the centrosome is gamma-tubulin and the gamma-tubulin ring complex, which can act as a nucleus, and in the centrosome is high concentrations of the tubulin dimers. So you will get nucleation and you will get polymerization in the centrosome because that's where all this stuff is. And that's how the cell regulates it, by regulating where the proteins are. So yes, you can get polymerization in other locations if you have high levels of the tubulin dimer in other locations, but usually it's not there, it's not translated there, it's only translated in certain locations, and the cell is regulating that. Make sense? Okay, so the cell is regulating that because it's trying to regulate the dynamics of the microtubules. I've mentioned before, microtubules are dynamic, they're constantly growing and shrinking. So in this movie, hopefully you can see the ends of the microtubules are constantly growing and shrinking into new locations. This is sort of a static image showing you the same thing. So there's a microtubule A that kind of gets longer and shorter. There's another microtubule B that kind of stays the same and then gets longer, and C seems to stay the same length. So different microtubules are doing different things based on their unique environment, and the cell regulates that environment. So if you're in the centrosome, you have a nucleating reagent, you have high concentrations of dimers, you're going to polymerize. As you start to polymerize, you're moving away from the centrosome. Eventually, you're going to polymerize, and the plus end of the microtubule is going to be in an area that's not above the critical concentration, it's below the critical concentration. There are no dimers wherever the microtubule has grown. So it's polymerized, and it's gotten away from the area of high critical concentration. So now it's below critical concentration. What's going to happen? Something called catastrophe, right? It's going to depolymerize. So if you're above critical concentration, you're going to assemble, you're going to get longer. If you suddenly get to a location where something occurs where you can no longer polymerize, the simplest thing would be you're below critical concentration, you're going to depolymerize. Other things can cause depolymerization. We'll talk about there's proteins that can cause depolymerization. But you're going to grow until the point where you can't grow anymore. Simplest way is because you have no more subunits. You're then going to depolymerize the assembly until a point where you're potentially going to be rescued. So you depolymerize, maybe you're in an area again now, you're above critical concentration, you're back close to the centrosome. So you're going to polymerize again until you get a different area, then you're going to depolymerize, polymerize, depolymerize. So when you look at the length of an average length of a microtubule, it's constantly changing. The average length, maybe for this one, might be somewhere like 20 micrometers, but it's constantly growing and shrinking. And as I mentioned before, that's important to its function because its function is transport. A cell doesn't necessarily want to transport something to the same location all the time. The cell is moving, it's changing. It may want to have exocytosis in one location, then it may want to exocytose in another location. So the microtubules are constantly rearranging themselves so the cell can do different things. Never mind that the cell can change shapes. So this feature is called dynamic instability. Microtubules are dynamically unstable, and they're designed to do that. They're designed to constantly be growing and shrinking, and that's important for function. And again, related to what's inside the cell, almost all of the microtubules are sitting with their minus ends in a microtubule organizing sensor. So their minus ends are kept. Gamma tubulin is sitting on that minus end, and there is no polymerization or depolymerization at the minus end. It's only the plus end that's growing and shrinking. So most of the functional dynamic instability is the plus end growing and shrinking, because the minus end is capped. It can't do anything. Okay, so let's look at this in more detail and sort of more complexities of this dynamic instability. This slide is sort of confusing to a lot of students. I'm going to try to go through it carefully and slowly and intelligently, hopefully. All right, so you're growing, and it makes sense. Once you get below critical concentration, you're going to shrink. But the cell wants to add to that dynamic instability. That's not enough. It doesn't want it that you're going to grow, and when you get to lower concentration, you're going to shrink. The cell wants to guarantee that this will shrink. It wants to guarantee that there's a mechanism put in that will make this thing even more unstable. And that mechanism has to do with the fact that that beta subunit can hydrolyze that GTP. If you remember, both of the alpha and the beta subunits in that dimer, both of them can bind to GTP, but the beta subunit can hydrolyze the GTP. Right? So you're making protofilaments. You have 13 protofilaments that come together to form this microtubule. Those 13 protofilaments have lateral cohesion. They like sticking to each other. Right? So all of these 13 protofilaments like sticking to each other. They stick better to each other, though, when that beta subunit is in the GTP form. So there's more lateral cohesion. It's more stability of all the protofilaments when the beta subunit is in the GTP form. Okay, the alpha is always in the GTP form. The beta can be GTP or GDP once it's hydrolyzed. When you have polymerization at the plus end, not only do you have to be above the critical concentration, but that beta subunit has to be in the GTP form. So if you're above critical concentration and that beta subunit in the GTP form, you're going to have extension. You're going to grow these protofilaments. Those protofilaments are going to have good lateral adhesion. The ones next to each other will stick to each other well. That's because that beta subunit is in the GTP form. So if you look at a growing microtubule, even though we don't know exactly how it grows, when you look at an electron microscope of one that's growing, all of those protofilaments are tightly stuck together. So if you look at the plus end, all those protofilaments are stuck together. There's stability there. Hopefully that will turn on in a bit, and hopefully no one else can hear it but me. So the plus n is always going to contain GTP during growth. Okay, so as you're adding subunits, all those subunits are in the GTP form. You form something called the GTP cap. Okay, so the plus n is made with the GTP cap, and that provides stability. Right, so you can then think of this microtubule as sort of a thick rope. Rope is made up of different fibers, and rope fibers are all tied together. If you have a piece of rope, and if you go to the middle of the rope and you try to pull those fibers apart, you can't pull the fibers apart in the middle of the rope. The middle of the rope is pretty stable. However, if you go to the end of the rope, you can start twisting apart the ends of the rope, the end part of it, and unfraying the rope, opening up the rope. And that's sort of what happens with microtubules. When it's growing and when things are stable, the plus end, the things are nicely stuck together. All the protofilaments have good adhesion. During disassembly, there is fraying. All the end pieces start coming apart. Okay? So the protofilaments have lateral adhesion. they stick together. That lateral adhesion is present in the middle of the microtubule, so it's difficult to put that middle of the microtubule apart. However, at the end of the microtubule, you can start pulling apart protofilaments, and that becomes easier once you hydrolyze that GTP. All right, so you have dimers, alpha beta dimers, both of them in the GTP form. They're above critical concentration. You're adding to the plus N. As you add into the plus N, you get this thing called the GTP cap at the plus N. The plus N is nice and stable and happy. And you keep on adding things to the plus N. However, once you add a dimer to the growing microtubule added to a protofilament, you actually start hydrolyzing that GTP in the beta subunit. So hydrolysis occurs by the beta tubulin after polymerization. So after you've added the subunit, you're going to have polymerization of that GTP. So when you look away from the plus N, if you're looking away toward the minus N, all the GTP has been hydrolyzed in the beta subunit, so all the beta subunit is GDP. So at the plus N is the GTP cap, but as you get away from the plus N, everything has been hydrolyzed to GDP. So that GDP is not stable. The protofilaments aren't as stable, but if they're in the middle of the protofilament, or sorry, in the middle of the microtubule, that's fine, it's okay. It's only when you get that GDP coming to the plus N. It's only when you eliminate that GTP cap. So when does this happen? Well, if you stop growing. If you're polymerizing, you're constantly adding GTP to the plus N, you always have a GTP cap. So if you're adding subunits, you're going to grow. That makes sense, that's what the cell wants. Once you get below critical concentration, you're going to stop adding a cap. Right? You're below critical concentration, you're going to stop adding a cap. You're below critical concentration, you're going to start depolymerizing in a second, because you're below critical concentration. That's not enough for the cell. It wants even more instability than just simply depolymerizing. Right? So once you stop adding, hydrolysis is going to keep on occurring. So hydrolysis is occurring after addition, so hydrolysis is going to keep on occurring until everything is hydrolyzed right up until the plus N. So now there is no more beta subunit in the GTP form. All of the beta subunits are in the GDP form, and now there is instability between those protofilaments and they start fraying. So fraying is occurring. So not only are you below critical concentration and depolymerizing, but you've gotten rid of the GTP cap because hydrolysis catches up to the plus N, and you start fraying as well. When you look at a depolymerizing microtubule, those protofilaments are coming apart as well as depolymerizing. Okay, so you have this double mechanism to make sure that depolymerization is occurring if this thing is dynamically unstable. When you get below critical concentration, you do not add any more subunits to the plus N. So the subunits that you've added that were in the GTP form eventually get hydrolyzed to the GDP form, and then you lose that stability between the protofilaments. A lot of words and a long explanation, but hopefully that makes sense. Otherwise, come see me during office hours or the forums, but hopefully that's clear. Okay, so there is a mechanism to allow for dynamic instability. The cell wants to regulate how stable things are. We can sort of study things using drugs. So there's a number of drugs that can both polymerize or stabilize microtubules. Depolymerize or stabilize microtubules. So you don't need to memorize any of the structures or anything, just the two drugs. So colchicine is a drug that you can add to cells. When you add colchicine, it depolymerizes them. Right? So you can depolymerize all the microtubules by adding a drug called colchicine. There's another drug called Taxol that stabilizes them. So basically they don't depolymerize. So if you add Taxol to a cell, all the microtubules will not depolymerize. Okay, Taxol is really complicated because depending on the dose, you can get prevention of growth and a variety of other things. But for our purposes, the microtubule is stable, it's going to stay the same size, it's not going to depolymerize. Okay, so one drug causes depolymerization, colchicine, the other drug, Taxol, stabilizes it, and you don't get depolymerization. Drugs like this, particularly Taxol, can be used as an anti-cancer drug. Okay, you need microtubule depolymerization during the cell cycle, and we'll talk about that later. Give me a second. And I'm back. All right, so there are drugs that interfere with microtubules, and those drugs will interfere with the cell cycle, and we'll talk about mitosis next lecture. So these drugs can be used as anti-cancer drugs, and indeed Taxol is a drug that's used in chemotherapy. But they're also very useful research tools because you can kind of then understand what microtubules are involved in if you depolymerize all the microtubules and see what can still happen, or if you don't allow them to depolymerize and that's what happens. Right, so we have drugs that can influence microtubule stability. Not surprisingly, these cells have proteins. There's a number of mechanisms, including proteins, that cells can use to either stabilize or destabilize microtubules. Unfortunately, most of these proteins are just called microtubule-associated proteins, called MAPs. So in general, they don't tell you what they do other than the fact that they bind to microtubules. Most of the MAPs that we're going to talk about are ones that stabilize microtubules. So in general, when I mention a MAP, it's going to stabilize microtubules. There's a bunch of proteins that we'll talk about that by definition are MAPs because they're binding to microtubules, but they actually destabilize them. Okay? But for stabilizing MAPs, what these are going to do is they're going to bind to the microtubules, they're going to coat the microtubules. So if you have a microtubule, MAP will bind to the surface of microtubules and not allow depolymerization. So it's going to stabilize them. Because the MAPs have certain structures and properties, it will allow microtubules to bundle. It allows microtubules to kind of come together in these large structures that can actually add function and strength to things. And that's most notable in nerve axons, for example. So if you look at this is cross-sections of two different nerve axons, you can see that there's microtubules in these nerve axons. These microtubules are kind of held together in these bundles. These microtubules provide stability to these nerve axons. So if you have a nerve axon, microtubules are used for transport. You don't want these microtubules to depolymerize, so they're coated with MAPs. Two important MAPs for stabilizing microtubules are one called MAP2 and another protein called tau. Okay, so these are found in a lot of nerves to stabilize the microtubules. The ones that stabilize microtubules have to have something called the microtubule binding domain over here. Okay, so part of the protein is a microtubule binding domain, and that binding domain will bind to the microtubules, and by binding to them, stabilizing them, and generally doesn't allow them to depolymerize. In addition to a microtubule binding domain, different MAPs have other different structures. In these examples, this MAP2 and this tau has something called a projection domain. So both of these MAPs have a microtubule binding domain, and both of them have a projection domain, but the projection domain differs in size. So a projection domain is just basically part of the protein that sticks out from the microtubule. But because these MAPs bundle the microtubule, they hold the microtubules together. Because they have different sizes of projection domains, when they bundle the microtubules, the spacing between the microtubules will be different. So MAP2 has a long projection domain. So if you look at microtubules that are coded in MAP2, they have this sort of, they're spread apart quite a bit because MAP2 projection domains kind of keep things apart. If you look at microtubules that have tau as a protein, the tau projection domain is smaller, so the microtubules are packed closer together. So using different microtubule using different maps, you are going to space the microtubules in different ways. This is going to give structural differences. So you're going to have stronger or weaker, depending on how you bundle them. The strength of those microtubule bundles are going to be different. And also what you can transport will be different. If you have lots of room in between the microtubules, you can move very large vesicles up and down. If you have smaller room, you can restrict movement of things. All right, so you have maps that stabilize microtubules, which is good, but sometimes you want to destabilize things, you want to get rid of things, so you need a mechanism to destabilize these maps, and that mechanism is phosphorylation. Right? Sometimes you want to make microtubules unstable, and for example, during the cell cycle, you guys have already talked a little bit about the cell cycle, and you will talk about it more perhaps. If you remember, you learned that cyclin-dependent kinases, in part of the cell cycle, part of their job is to phosphorylate MAPs. So when you phosphorylate MAPs, they let go, they release their microtubule binding domain, and then the microtubules can depolymerize. So MAPs are involved in stability, but they can, through phosphorylation, be removed from microtubules. So some maps, their function is relatively straightforward and easy to understand, stability and sort of packing. There's many other maps present, and a lot of them we don't really understand exactly what they're doing. There's a number of maps called plus tip maps. As the name suggests, they're found at the plus side of the microtubule. And when you look at their location of things, often we see that when the plus tip map is present, the microtubule is growing. So here it is showing you over time the presence or absence of this protein called a plus tip map. If in green is the presence of a plus tip map. So if the protein is present on the plus end, it tends to grow. If the protein is not present, it tends to shrink or get deassembled, depolymerized. So plus tip maps seem to help promote polymerization by stabilizing that plus end. So there's a number of these proteins, and it looks like, yes, they do stabilize the plus end to help things. However, when you look at the kinetics of these proteins, particularly this one protein called EB1, EB1 is present at the plus tip, it's present on the plus end, but as you can see when looking at the protein, it's also present on other parts of the microtubule, and it seems to actually move along the seam of the microtubule. The seam of the microtubule is where the alpha and beta dimers don't meet, right? So the important part is that EB1, this plus tip protein, does stabilize the plus end, but it also seems to be able to move and perhaps take cargo to the plus end of the microtubule. So there's a lot of proteins that we're just not quite sure what their function is. They certainly involved in stability of the plus end, a bunch of plus tips, but EB1 is possibly involved in both stability but also in transport, perhaps. Okay, so you have maps that stabilize, plus tips that also seem to stabilize. Not surprisingly, there's sort of other things that can also promote stabilization or destabilization. Okay, so two proteins that are also found at the plus end involve a domain called the TOG domain. So TOG domains are present in a protein called XFAP215 and a protein called CLASP. They bind to the plus end, and they can stabilize the growing plus end, but they can also promote it by being removed. right? So if you have these proteins present, you have stability, but if you remove them, you're going to have more likely catastrophe. Okay, so proteins with a Tog domain are going to bind to the protofilaments at the plus end, and if they're present, they're going to stabilize, but if they're absent, they're going to kind of promote catastrophe. Simple enough? Just memorizing that XMAP 215 and CLASP have Tog domains, and they can stabilize the plus end by binding to individual protofilaments. There are also proteins that cause destabilization. They basically are going to promote disassembly. One of them is a protein called kinesin 13. Kinesin 13 uses ATP hydrolysis, so it basically uses energy to literally rip off dimers off the plus end. So in the presence of kinesin 13, in the presence of ATP, you're going to use that ATP hydrolysis to pull dimers off the plus end. So if you have enough kinesin 13, if you have enough ATP, it doesn't matter what the critical concentration is, this can overpower things and cause depolymerization if you have enough of the kinesin 13 molecule. Okay? It can also work at the minus end, but you have to consider that the minus end is always capped, or almost always capped, by gamma tubulin ring complex. So kinesin 13 uses ATP hydrolysis to basically rip dimers off from the plus end and promote depolymerization. Another protein that promotes depolymerization is a protein called staphmen. Staphmen, when it binds to the protofilaments at the plus end, basically promotes GTP hydrolysis. So it's getting rid of that GTP cap. So it's causing fraying at the plus end by promoting the protofilaments coming apart. So in the presence of staphmen, the GTP cap is potentially eliminated, so therefore you have GDP in the beta subunit, and you have poor lateral adhesion from the protofilaments, and it will promote fraying and then depolymerization. Fun? All the stuff that the cell can do? Okay, so that's sort of the basics of microtubules. What they really do, though, microtubules, is move things. So, moving into section 6, basically, or the next lecture. Microtubules are basically tracks or roads along which things move. And we have seen this for a long time. One model that's been used for decades is squid axons. So squids are aquatic animals. They have very long sort of tentacles, and within those tentacles are axons, and you can remove those axons and you can exude all their cytoplasm. So you can take a squid axon and kind of squish out all their cytoplasm, and in that cytoplasm are microtubules and everything else. But you can then look at things moving around. So when you look at these sort of preps of squid axon cytoplasm, you can see microtubules depending on how you do the preparation, but you can see material moving along microtubules. You have large things moving on microtubules, small things, and things are moving in both directions. Again, depending on the preparation that you do, you can see the microtubules and you can see different things are moving at different speeds in different directions. And indeed, you can see that in other levels. You can do electron microscopy, and you can see different types of organelles that appear to be bound to microtubules. You can do all kinds of light microscopy where you can see microtubules, and you can see particles moving along those microtubules in both directions. So the microtubules look like they're tracks for things moving along or roads, and it looks like something is moving along those microtubules. Right? In trying to figure out those things, people wanted to isolate the proteins that are doing this. So this experiment was carried out to find these motor proteins, these proteins that are moving along the microtubules. Okay, so again, this is using squid axon as the model. This is not what a squid axon looks like, but regardless, this is an axon. So imagine you have an axon along squid axon, and you have the axon body. So the axon body is where translation is going to occur. So you have the main part of the cell body, that's where translation is going to occur, and into that cell body you inject radioactive amino acids. So you put in radioactive amino acids as translation is occurring. So now translation is occurring and you're making radioactive proteins. All the proteins that you're making are radioactive. Then you wait a certain amount of time after you've injected these radioactive amino acids, and now you have radioactive proteins, and this is an axon, a squid neuron, there's an axon available. Proteins are going to be transported in different directions. Some of those proteins are going to go into that axon. So you inject amino acids, proteins are made, and then proteins are going to be transported into that axon. So what you're going to do is do an experiment where you do injection and then do different time points and isolate proteins from axons at different time points. So using different axons. So you take one axon, inject radioactive amino acids, wait 5 minutes, and then isolate protein from different parts of this axon. So in the first experiment, for example, here they waited a certain amount of time, let's say 5 minutes, and they isolated proteins from 4 parts of the axon. So the part of the axon closest to the injection site, next part, next part, next part. So you're isolating proteins from these different regions at different time points. And then you run those proteins on a gel. Those proteins are radioactive because you injected radioactive amino acids. So after you run them in the gel, you expose this gel to X-ray film, or you do some sort of imaging so you can see the radioactive molecules, and you get bands. So this is an SDS page. You get protein bands that represent the proteins there. So these are all the different proteins, the smallest proteins on the bottom, the largest proteins at the top. So you have a certain banding pattern. There are no colors because there is no colors with radiation, but you can put colors on things because you start seeing things over and over. All right, so you injected radioactive amino acids, you wait a certain amount of time, isolate protein from 4 segments, run them on a gel, and you see a certain pattern. Now you do this again with a new axon. You inject radioactive amino acids, wait a certain amount of time, now you're waiting 10 minutes. Last experiment was 5 minutes, now you wait 10 minutes. And then you take 4 different segments, and now you isolate the proteins from these different segments, and you run the proteins from these different segments on a gel. So in the 4th segment, you run it, you get a certain banding pattern. So you have the proteins from the 4 segments, you run them on the gel, and again, you image them. There is no color, but you can see by the molecular weights of the proteins that they look similar. So you see these 3 proteins here that are a certain size. Excuse me. These proteins that were in segment 4 used to be in segment 2, right? And these proteins that were there here in segment 2 used to be in segment 1, so you kind of color them the same to kind of show you that they are moving together. Now you do this experiment again. Take another squid axon, inject radioactive amino acids, now you wait 15 minutes. So now when you isolate the proteins, these red proteins after 15 minutes are gone. So when you isolate the next time point, the 4 segments, and run them on a gel, there are no bands that look like those red proteins. Now you have different proteins coming on, represent proteins that are moving slower into the axon. You do that again now. You do another time point, inject radioactive amino acids. Now you wait 20 minutes and isolate proteins from these different segments and run them on the gel. So what is this telling you? It's telling you 2 or 3 things. One is that proteins are moving together at the same speed. So these 3 pink proteins are moving at the same speed. These blue proteins are moving at the same speed. These purple proteins are moving at the same speed. Right? Potentially they're moving as a complex. This is also showing you that different things are moving at different speeds. These pink proteins are moving as a group faster than these blue proteins, which are moving as a group faster than these purple proteins. So this movement isn't just random. This movement seems very specific at a certain speed, and certain proteins seem to be working together. So what you can do then is go into this acrylamide gel and actually pull out these protein bands. You can isolate these protein bands and sequence them. Back when this experiment was done, that was very difficult. Now sequencing proteins is pretty easy. But what you can do is isolate these bands and sequence them and see what protein is it. And this is how they discovered the motor proteins, particularly a protein called kinesin. Okay, so kinesin is the plus N directed motor protein for microtubule that's moved from minus N to the plus N. There are many kinesin genes and classes of genes, okay, and they're involved in different types of transport of different types of cargo. Okay, so there's lots of variation in different kinesins, perhaps not surprisingly, because they're moving many different things. There's general characteristics I would like you to memorize to kind of cover, in general, what kinesins are. So, in generally, there's 2 kinesin heavy chains and 2 light chains. Okay? So, this is sort of an artistic representation of kinesin with heavy chains and light chains. This is sort of a more 3D structure of the heavy chains. Okay? So, later when we do dynein and then later when we do myosin, all of these motor proteins have certain structures that are similar, but the names vary. So I'm going to start or try to stick to one specific naming, and it will try to be consistent with things. Okay? So the heavy chains basically have a head domain, a neck domain, and a tail domain. So there's a head, a neck, and a tail. The head can move on the neck, so the neck is flexible, and the head moves because of the neck, and then there's the tail region. You can see here that the tail is also called the stalk, and the tail is at the end, but let's just stick to head, neck, and tail. So the heavy chain of kinesin is made up of a head, neck, and tail domain. This head domain has an ATP binding site. So it's an ATPase. This head domain is also the microtubule binding domain. So it's the head domain that binds to the microtubule. It's the head domain that uses ATP hydrolysis to move towards the plus N. Okay, so kinesins have heavy chains and light chains. The heavy chain has a head domain, neck domain, and a tail domain, and it's the head domain that binds to the microtubule and moves along. On this tail domain of many kinesins is the light chain. Often there are 2 light chains. So the light chains are what bind to the cargo. Okay, so the heavy chain has a head domain that binds to the microtubule. The other end of the heavy chain is the tail. It will bind to light domains. There are different types of light domains, so they're variable. Those light domains bind to different types of cargo, and then they move that cargo towards the plus N. Okay, so this is sort of the simple explanation. A heavy chain that has the head domain and the light chain that binds to the cargo. So far, so good. There are many types of kinesins, 14 classes, 45 genes. You need to memorize 4 of them. All right, so there's kinesin 1, kinesin 2, kinesin 5, and kinesin 13 are the ones that we're going to focus on in this class. Kinesin 1 is the most abundant one, as it says up here. It's conventional. I won't say most, but a good chunk of the kinesins in your cytoplasm are kinesin 1. Those are the typical kinesins. They have 2 heavy chains and 2 light chains. Right? The heavy chains have the head domain that binds to the microtubule. The light chains at the tail end bind to the cargo. Kinesin 1, the 2 heavy chains are the same. Right? So they're homodimers of the heavy chain. Okay, so kinesin 1 is the most abundant, kinesin 1 is the most studied, kinesin 1 kind of makes the most sense. Kinesin 2 is also important in all kinds of transport, particularly in cilia and flagella, which we'll talk about. It is heterotrimeric. It has two heavy chains, but they're not the same. And it has another protein that's almost like a light chain. It's basically a kinesin-associated protein, but we can call it a light chain because it is basically does the same thing as a light chain. So kinesin 2 has three different proteins, two heavy chains, and something like a light chain. So again, the head domains are going to bind to the microtubule, and the light chain is going to bind to the cargo, and it's going to move to the plus end. Okay, so if you can imagine, if you're doing an experiment and you're looking at kinesin 2 is going to be moving to the plus end. So in that experiment where we talked about, oops, wrong way. In this experiment where we talked about, you have a protein that's moving towards the plus end, and then there's three proteins that are together, moving at the same rate. So perhaps, you know, something like these pink proteins could be something like kinesin 2. It has two heavy chains that are different molecular weights, and then a light chain of different molecular weights. So these different banding patterns are showing you the different types of kinesins and different sort of configurations and different binding partners. So it's easy to imagine kinesin 3 would give you three bands because it's heterotrimeric. I should have mentioned here, most kinesins are plus end directed motors. Some do strange things and can also go to the minus end, but for the purposes of this class, if I say kinesin, it's moving to the plus end. So kinesin is moving to the plus end unless I specifically say otherwise, but for our purposes, kinesin is moving to the plus end. Okay, so kinesin 1 and 2 are the main sort of kinesins that are moving cargo, and they have these light chains that recognize different types of cargo. Kinesin 5 is a little unique in that it's only form of heavy chains. There are no light chains. Kinesin 5 is made up of 4 heavy chains that come together in this structure. So you have one pair of kinesin 5 molecules and another pair of kinesin 5 molecules that come together in this bipolar structure. So their tails overlap. So you have two head domains on one side, two head domains on the other side. Their tails are overlapping. They're not binding any light chains. They're not really transporting any cargo. So you have 4 kinesin 5 molecules that have head domains on both sides. Bipolar means both sides are identical. You can flip this over and it looks the same. Because they have head domains on both sides, they can bind parallel microtubules, and they do. So you can put kinesin 5 in between parallel microtubules, and the head domains will bind to the microtubules, and then the head domains on either side will try to move towards the plus end of the microtubule. When that happens, the microtubules slide apart, and we'll talk about this later when we talk about mitosis. So kinesin 5 is involved in microtubule sliding. It pushes microtubules apart. And we'll see how that works when we talk about mitosis later on. Okay, so 1 and 2 is involved in transport. Kinesin 5 is involved in microtubule sliding. Kinesin 13 is involved in depolymerization, in disassembly. We've already talked about it when we talked about instability. Kinesin 13 is basically 2 head domains and neck domains. It doesn't have a tail, it doesn't have light chain. It's not involved in transporting anything, but it uses its ability to bind microtubules and use the hydrolysis of ATP to basically rip apart microtubules. So it's not moving along microtubules, it's disassembling microtubules using its ability to bind to and hydrolyze ATP. Okay, so you got kinesin 1, 2, 5, 13. kinesins move along the microtubules towards the plus end in general. They do so using ATP hydrolysis. The way that ATP hydrolysis is used is explained on the right. Ignore it for now. Later on, we'll do myosin. Myosin and kinesin kind of do the same thing. When we talk about myosin and actin, we'll go through how ATP hydrolysis is involved. More importantly, what I want you to take away from this is we know how far things move. All right, so upon ATP hydrolysis, if each head is moving 16 nanometers. Okay, so when it's moving, as you saw in the movie we watched, and we'll see in other movies, the heads move past each other, so the heads are moving like you would be walking. Okay, so if you can imagine, if the two heads, if you're not walking, the two heads are 8 nanometers apart, but when it takes a step, the back head is going to move in front of the other head, and it's going to move 16 nanometers. So the movement of each head is 16 nanometers, but the space between the heads as it's moving is 8 nanometers. Another thing to consider is you don't want these things moving, especially the ones that are transporting cargo. You don't want kinesin 1 and kinesin 2 transporting, you don't want them moving to the plus end if there is no cargo. So kinesins in general are inactive, they're folded and not able to bind to the microtubule in the absence of cargo. So in this example, you have kinesin 1 here. If there is no cargo, the light chains can't bind to the cargo. So with no cargo, it can't bind to the microtubule. In the presence of cargo, the light chains bind to the cargo. Then it opens up the kinesin. It allows the kinesin to bind to the microtubule. And then the kinesin can move using ATP and 16 nanometer step towards the plus end. Make sense? Okay, so we're going to talk quickly about dynein, and then we'll start again with this in the next lecture. The biggest annoying thing here is the artwork and the name of things. So dynein is the minus end directed motor for microtubules. So kinesin takes things to the plus end, away from the centrosome. Dynein takes things towards the minus end, towards the centrosome. Dynein is very similar. It has a head domain, a neck domain, and a tail domain, except they've drawn it differently and they've called things differently. So try to ignore the differences in the colors and the structures, right? So it has a head domain. So here in red is this head domain. The head domain binds microtubules. The head domain uses ATP, except this time it moves toward the minus end. It has a neck domain here called a linker, and it has a tail domain here, it's called the stem. So it's still the same structure, head, neck, tail, slightly different name. Even more annoyingly, in the middle of the head domain is this thing that pokes out, and they call it a stalk. It's still the head domain. It's just that the head domain has this extra structure that actually binds to the microtubule. But don't let the shape fool you or confuse you. There's a head domain. The head domain binds to the microtubule. There's a neck domain, this linker that's flexible, and then there's a tail domain that's going to go towards binding the cargo. Okay, and we'll start here again on Wednesday. Thanks. It's a cheap thing from Amazon. It's a wrapper. It just looks cool.
Oh, it's on hand, bro.
No, you have to wind it.
Oh,
Yeah. See the stuff moving inside there.
yeah, I like it. It looks nice.
Like it. What's this thing in the quick function, but then go for it.
What do you think? Like I know they're open book, but I feel like it still requires like